Thank you. I'm going to talk about the sickness of evidence-based medicine. I don't believe medicine should be based on evidence. Now that sounds ludicrous. What I think is we should have medicine based on science, rational medicine. And to do that, it's relatively straightforward. All we need to do is let patients use their common sense. So what I mean by that is we want to have the concept of a rational patient and a rational doctor. And by rational, well, rational is just a posh way of saying somebody who uses their common sense. There was a lot of controversy before this conference, and I was going to explain why we asked Professor Ernst to come and debate the issues about EBM, but given what has transpired earlier, I don't think I need to give that explanation at all. Professor Ernst was asked would he like to come along and explain why people doing alternative medicine or complementary medicine or orthomolecular medicine or ecological medicine should be doing EBM. Of course, he was busy, which we predicted he would be. And why did we predict he would be? Because we've been trying to get somebody to debate these issues with us for some time. We issued a $100 trillion prize for medicine. These were Zimbabwe dollars. That was to illustrate how EBM has devalued the currency of medical science. We had cartoon videos on YouTube, and so far we have had no takers. Nobody wants to debate the issues of EBM. Nobody wants to explain why anybody should be using this form of social science instead of real science. What's wrong with EBM? EBM, if you're a mathematician, does not make sense. If you're a decision science, it does not make sense. If you're an experimental scientist, it does not make sense. Like the Queen, if you want to believe in EBM, you have to start by thinking 6 impossible things before breakfast. So if we have been able to inspire one of these skeptics to take up the challenge, what would we have said to them? Well, the first thing we would have told them is a randomized clinical trial has no special properties. It's just a very poor experimental technique. It's weak. And the weakest form of these clinical trials are the large ones. Just imagine you wanted to replicate, to check whether one of these large-scale trials was correct. How could you do it? Have you got $50 million to repeat this trial? If you want to test whether Isaac Newton was right, all you have to do is drop an apple. And if it goes up or sideways, then you know his ideas about gravity were not quite right. The power of science is being able to repeat the experiment. So a small, simple experiment is better because you can check it. This is a typical example from an EBM clinical trial. I could have picked a real one, but it doesn't matter. They're all the same. They will tell you that 30% more people benefited from a painkiller. Could be 20%, could be 50%. But what they don't tell you is in reality this was a large trial, and 3 people in 1,000 actually obtained benefit. Now back to that rational patient. Imagine walking in to your doctor's surgery and asking him, what is the chance of you benefiting from the treatment? If they tell you 30%, you think, well, 3 in 10, that's quite reasonable. But if they tell you 3 in 1,000, you might say, well, is there anything better than that? So these large-scale, randomized, placebo, blah, blah trials just measure unimportant differences in populations. And the problem is, what a population does not apply to a single individual. We had an election a short time ago, and the Conservatives won. But I can't point at any one person in this audience with any degree of reliability and say, you voted Conservative. I don't really care what you voted, but whether that's right or not, I wouldn't want to put a bet on it. You cannot apply results from comparing groups, especially large groups, to individuals. The mathematics doesn't work. Placebo-controlled trials. All these trials, except those for cancer chemotherapy, are supposed to be placebo-controlled. You can ask yourself why a highly toxic drug they don't want compared with a placebo. But the other thing about it is a placebo-controlled trial is unethical. That's the World Medical Association. The only people who like placebo-controlled trials are the Food and Drug Administration and pharmaceutical companies. Sorry, but they're the people who are arguing against, arguing for the misuse of placebos. Consider you going into, you look an affluent audience, a Mercedes garage to buy a new car. And they say, we've got this wonderful new sports car, and we've proven it's effective. And you ask them how they've proven it's effective. Well, we sat somebody in this chair, the placebo, and we sat somebody in the car. And the person in the car could go faster than the person sitting in the chair. And it was much more likely to get to the destination. EBM promotes drugs that work slightly better than absolutely nothing at all. We had a look at meta-analysis. We looked at 2 years of reviews from Cochrane and Jarman, and not one of the reviews was valid. Not one. The biggest problem was selection. These were so-called systematic reviews. And the way the rules played was the researcher could say they were studying vitamin E, and they wanted vitamin E to not be beneficial for heart disease. It's easy. They just pick the studies that are least beneficial for heart disease and find a way of excluding, a rule for excluding the studies that are good for heart disease. And they can impose that rule themselves. If they wanted to show it went the other way, all they had to do was use a different rule. This is not a systematic review. It's a statement of the prejudice of the person conducting the review. And they hide that by describing it as being subjective. We asked Cochrane to defend themselves. We're still waiting for their explanation of how these meta-analyses can be at all valid. So far, they haven't explained it. So a rational patient, that's a patient using common sense, would want to know the chance of a treatment helping them personally? A rational doctor would want to know the chance of a treatment benefiting this patient, the one that is sitting in front of them, personally. Now, evidence-based medicine works on groups of individuals, especially large groups of individuals, and very, very tiny effects. The chance of a drug so proven by evidence-based medicine helping that patient is minimal. It's not designed to do that. The experiments are to tell the difference between large populations. So are there other ways of doing it? Well, yes, you can use Bayesian statistics, or you can use pattern recognition, or you can use game theory, or there are a whole series of techniques called decision science. Now, as I said before, I've got to cover several things in this talk, and the linking thing I'm using to link all of these things is common sense, or if you're scientific, rationality. Now, the medical position on vitamin C is also irrational, but it's easier to see. Now, the National Institutes of Health claimed about 20 years ago that the body was saturated at an intake of only 200 milligrams. That's important. And it's important because it means if you go down to your health shop and you get some vitamin C tablets, standard 1 gram tablets, the dose is too high. You're just wasting your money because you'll only absorb 200 milligrams of it. The other 800 milligrams, you may as well not buy. You could just go and get a 200 milligram tablet, and that would be the maximum you could absorb. This is vitally important because these results are vitally important. That claim is important because the RDA for vitamin C is specifically based on their results. These are their results. The lowest graph here with the round circles on are their results, plasma levels for 200 milligrams of vitamin C for 24 hours after a single dose. Now, as you can see, that saturates the body, and all these other data points are below this line. Sorry, I find it difficult to relate this because I always want to burst out laughing. Okay, this is low, that is high, and these are above that. Okay, the square boxes filled are for 1 gram. And as you can see, every single point of that 1 gram dose is above this 200 milligram dose. Now they say this dose saturates the plasma. So there's no point at taking any other dose. Now these are single doses from 1 to 3 grams, is it? Yes, 1 to 3 grams. And you can see how they decay. How did they come to this conclusion? They gave a dose of vitamin C and they waited until it had been excreted. Hours and hours, 12 to 24 hours. You couldn't quite pin them down on it. So they gave a 1 gram dose over here. They would wait until it had all been peed out and then measure the blood levels. Vitamin C has a half-life in plasma at about 30 minutes. This is their data, it isn't my data. The two top graphs, which are sustained levels are for roughly 10 grams and 18 grams each. That was 20 years ago. The National Academies of Sciences described this study as uniquely rigorous. We told them about 10 years ago, and they couldn't explain how this had happened. The best response we got from them was, "We don't understand." Now, unfortunately, even though this was made very public at the time, it was an embarrassment to the NIH. Studies are still coming out saying the body is saturated with 200 milligrams of vitamin C, and probably the next RDA will be based on saturation. For example, Bowles Fry, head of the Linus Pauling Institute, has recently published a paper suggesting a new RDA for vitamin C, and he's claiming it should be 200 milligrams because the body is saturated at 200 milligrams. This is the head of the Linus Pauling Institute, which originated in part to study high doses of vitamin C. Go figure, as the Americans might say. They claim that oral doses can't produce anti-cancer plasma levels, and they pushed very heavily for IV vitamin C as a treatment for cancer. The trouble with that is their own data show that the oral doses of vitamin C, even their low levels of vitamin C that they've achieved from oral doses are sufficient to act as an anti-cancer cytotoxic agent in some cases in their experiments. This is their data. Our data is quite different. It doesn't top out at 200 micromoles per litre. It's 600 or 800 if you use liposomal vitamin C and normal cheap ascorbic acid in combination. And I suspect you can get to about 1 millimole sustained indefinitely. But I haven't shown that. We've just got close to it. Now, oral doses are preferable for fighting cancer. That is, you would not choose IV vitamin C over oral. But let's go into that a little bit slowly. Okay, cancer's adaptive. What does that mean? It means that a cancer cell will try to survive if you try to treat it. It's not like some normal cells which it's easy to induce them to commit suicide. Overall, the tumour will fight back. And it'll use any method at its disposal to overcome your treatment. If you block a particular gene, doesn't matter what gene, just pick one, the cancer is not a single clone. It's more like an ecosystem. Think of it more as a tropical rainforest. It's that variable. If you count the number of chromosomes in cancer cells from a single tumour, they will go from 10 to over 100. Not necessarily in every tumour, because they will vary, but 10 to 100 would be quite reasonable. They're massively genetically different. So if your treatment hits one gene and affects half of the cancers, the other half of the cancers may not even have that gene. The way I want to try and present this is in terms of treating bacteria, bacterial infections. And the bacterial infection that I think of is TB. To treat TB, classically you would give perhaps 5 different antibiotics over a period of 6 months, and you would give them continuously. And the reason for that is it's a slow-growing, difficult-to-treat infection. And as you're treating it, some of the cells may become resistant to 1 or 2 of the antibiotics. And then, at least initially, it would still have 3 or 4 antibiotics still to overcome. And by treating it with a barrage of different antibiotics, which operate using different mechanisms, the TB can't adapt as easily. At least it didn't used to be able to. Antibiotics fail because the bacteria adapt, and normally that's called resistance. Chemotherapy and radiation fail for essentially, in principle anyway, the same thing. The tumour adapts. Except that typically a tumour adapts rather quickly to chemotherapy and radiotherapy. So we'll think about what's going on, but as we do, before we go on, we should say shrinking tumours is easy. By easy, I mean there are thousands of substances out there that will kill cancer cells and leave normal cells unharmed. Somebody asked about curcumin this morning. That's an example, but there are thousands of them. And every so often, like in this case from the University of Alberta, somebody in academia finds a new substance, in this case an old drug, that will kill cancer cells and leave normal cells undamaged, relatively speaking. And they think they've made a massive breakthrough. Unfortunately, they're working with the current paradigm of medicine, in which chemotherapy needs to be really macho. There's no way in which you can really approach the treatment of cancer through chemotherapeutic methods that isn't harmful to the patient. Well, it just isn't true. There are thousands of these substances, but the problem is any substance that you use, any one of these that you try, the cancer will rapidly adapt to. So, supposing you're a biologist and somebody comes to you with a petri dish of some bacteria and says, my antibiotic will not kill these bugs. Can you arrange for these bugs so that my antibiotic will kill them? What might the biologist do? Well, one recipe for it, or an algorithm for it, would be to treat regrow the culture in a different petri dish to begin with, but treat a sample of the bacteria with a low dose of the antibiotic, perhaps killing half of them, and then wait a little and then apply the same antibiotic and killing half the remainder, letting it regrow somewhat, and then kill half of the remainder. It would be a process of treat, wait, and a cycle like that. You don't get doctors asking you to take penicillin on Mondays of each week for the next 6 months for the same reason. The trouble is, chemotherapy and radiotherapy must be given intermittently in that way because they're so toxic. Okay, so chemotherapy and radiotherapy, I'm being a little bit facetious, but there are mechanisms for making the tumour untreatable, because it will adapt to those treatments and by default other treatments, because many of the treatments work in a similar way. The primary question in cancer treatment is how to stop the cancer adapting. If you think about it, that is the core problem. Once again, the cancer will try to adapt to any treatment. It will get round, if it can, any pathway you block to slow its growth or try and kill it. The approach that is likely to be effective is like treating TB. You block multiple mechanisms all at the same time and keep that as sustained pressure on the tumour. One of the problems with nutritional treatment of cancer is the idea that you feed the person to strengthen the person to fight off the tumour. Now that's a dangerous approach, because that tumour, those cells in that tumour only have the same biochemistry as your normal cells. They haven't got any new proteins or new enzymes or anything like that. And essentially, those same nutrients that build up the patient are often growth factors for the tumour. But there are plenty of growth factors, nutrients that are growth factors, and most of you could probably add many more. Conversely, if a tumour cell is short of a nutrient, it is very difficult for it to grow. In order to grow and divide, a cancer cell has to build a complete copy of itself. So it needs the materials for producing new DNA, new proteins, new structures, new lipids. The whole cell has to be duplicated. So if it hasn't got, for example, enough sulfur-containing amino acids, or just plain sulfur for argument's sake, it cannot actually duplicate itself because it needs that in order to go through the process. So, in principle, depriving the tumour of a nutrient is an effective way of stopping it growing. But it will try to adapt. The one case you can think of that it couldn't adapt is if you deprived it of energy, because from basic physics, it's not going to be able to generate its own energy. Shortage of several nutrients can block several pathways all at the same time. And if you block sufficient pathways or deprive it sufficiently, it's not going to be able to grow. Earlier we've heard about antioxidants and blocking genes and also killing cancer cells, and Damien presented some nice clinical data on that, which I'm very pleased about because I'm going to cover it in a mouse study. And it's a historically classic mouse study. The Linus Pauling Institute was formed by Art Robinson and Linus Pauling. Art Robinson did all the fundraising and the management and the organisation. And they started an experiment with mice and vitamin C. And this generated an enormous amount of controversy and a big public argument between Art Robinson and Linus Pauling, and they split up. It was very acrimonious. Now the problem was Art Robinson started this experiment on mice, and he started by giving them about 3 grams a day. When I say 3 grams, I don't mean the weight of a mouse. I mean 3 grams in human terms, the equivalent, the mouse equivalent of 3 grams of vitamin C. And what he found was the mice given the supplement died more quickly. Their tumours grow more quickly. And so Art Robinson said, we're giving advice out for people to take supplements of vitamin C, and the data we're getting from the experiments suggests that it's harmful, so we should change our advice. And Pauling said, no, we shouldn't change our advice. We should wait until we've got the full data. It turns out both of them were right, but for different reasons. Here's the study, how the study panned out, because Art Robinson continued the study after he'd left the Linus Pauling Institute. And what he actually found was if you gave a great deal of vitamin C, the mice tended to live a lot longer and had far fewer tumours. So in this case, we've got the controls, and 90% of them had minor lesions, and 1 in 4, 1 in 5 had major lesions, with a human equivalent of 192 grams of vitamin C. No mouse after 2 months had any major lesions, and only 16% of the mice had minor lesions. Now, Art Robinson also had an idea about nutrient depletion, in which instead of giving supplements, you deprive the mice of nutrients. And he did this because people were following the health suggestion that cancer patients should eat raw vegetables so they could lose a lot of weight and have very few nutrients to help the cancer grow. And in this case, raw vegetables, giving raw vegetables is not to make the mice healthy vegetarians. It's to slow the growth of the cancer and to almost starve the patient. And when he did this with the mice, he found that the results were almost as good as very high dose vitamin C. As you can see, only 1 had major lesions and 11 out of 40 had minor lesions. And combining the two was even more effective. There were no major lesions and there were even fewer minor lesions. One of the problems is people picked up on the raw vegetables and thought, well, we can do better than that. We can juice them. And juicing is great. It's a good way to get a lot of nutrients in the system, but that's just it. It turns your raw vegetables, which are very difficult for the mice to, in this case, the mice or humans to digest, into a glass of sugar with added vitamins and nutrients, which is the opposite of the original regime. So, what am I trying to say? What I'm trying to say is that all these calls for evidence-based medicine are misleading. They're not calling for science-based medicine or rational medicine. They're calling for evidence-based medicine. And if you track it back, you'll find that their evidence hierarchy was not determined by scientists. It was determined by the US Supreme Court on what evidence should be acceptable in a court of law. So in other words, lawyers were telling scientists how to solve problems. Even the lawyers themselves had, even the judges, shall we say, Supreme Court judges themselves, two of them, objected to the ruling on the grounds that they didn't understand what falsification was, i.e. showing something wasn't correct. And they were not at all sure that any of the standard judges would either. So the question is, do you want to be treated by doctors using legal medicine, or do you want to be treated by doctors using scientific medicine? Because the two things are totally different. To make medicine scientific, you do what the decision scientists do. A patient does what is in their best interests. They're not interested in what's best for the population as a whole, in principle. They're interested in getting a treatment that is safe and beneficial for them. The doctor is interested in giving that patient a treatment that will work for that patient. And then the questions you ask is, what is the chance of this treatment helping me? Now, that's only for one patient. But you can get that data. And if you then sum up all these individual patients, you actually get an optimal result. You get just about the best result for the population that is possible. What you can't do is say what's good for the population as a whole and say that's good for you. It's the standard old joke. The average person has exactly one breast and one testicle, or 1.95 legs. Yeah? Okay, with that, I'm going to end this talk, but it's just with one statement. Don't be afraid of people who are claiming that you should be evidence-based, because there is no value in that. Has anybody here seen a validation of a randomized clinical trial, the method of randomized clinical trials? Has anybody seen it compared with, say, game theory or pattern recognition or artificial intelligence? They claim EBM is a gold standard by authority. They just say it's a gold standard. There is no supporting data. Notice I'd try and avoid saying evidence. The best thing for patients is they start to actively look after their own decisions and demand the information that they need to make those decisions. And the best thing that doctors can do is help the patients or guide the patients individually in what is best for them. Thank you very much.